So getting started with the GDL. You download and install the desktop loading application and then connect the loader to the database and file management stack. So you log into the loader UI by entering the web service URL, your user and password. Upon login, you are presented with several panes on the left. Using the loader UI, you can create new projects, experiments, data. For more information about each of these, you can find document. And lastly, the wizards, which are used to load markers, germplasm samples, and data sets. Activities that will be performed more frequently are at the top of the list. The items with blue stars must have some information before the data can be loaded. We deploy GOBii with predefined properties, which we call controlled vocabulary. CV terms may be provided with the system or generated by the curators. System terms often play a role in the functionality of some of the applications. Defining organizations. These can be user affiliated, genotyping service providers, research centers, research labs, private companies, etc. You will need to enter the organization's name, address, and website, and add new. You can also update an existing organization. Defining context. You need to select an organization. You need to enter last name, first name, username, and email, and then select a role for that content. Again, you can add new or update. You can define platforms and protocols. Platforms describe different genotyping technology, for example, CASP or sequencing. Protocols provide additional details about the genotyping service and vendor. Defining analysis. Calling analysis is required for all data sets. There is also cleaning, imputation, and allele sorting. Each analysis can contain optional information about the software and the parameters used. This is an example of defining an analysis. You can add new or update. In the next pane, the create, you can create Mapsets, either genetic or physical, which are composed of chromosomes, linkage groups, scaffolds, and contigs. You can create projects, experiments, and data sets. There are some required fields when creating these entities. For example, creating a data set, an experiment, a data set type, and calling analysis are all required fields. The wizards are used to add basic metadata, load the data file, preview data from the file, map data fields, review and submit, and receive an email. These are the sections of the wizards where you add basic metadata, load the data file, and preview data from the file. On this page, we map the data fields. On the left will be displayed the column headers in the file you uploaded. These column headers are mapped to key fields and to additional properties. On the last page, you review what you are submitting and then submit by clicking Finish. Completed, you'll receive an email, either a success email or a failed email. The email will include a link to the error log to help you diagnose the error. Let's first walk through the marker wizard. In the preview data pane, you see the column headers displayed from your file upload. The column headers seen in the preview pane are those displayed on the next page under field headers. These are then mapped to the marker information table. Your success email will tell you how many markers were in your file, how many were loaded, how many have been previously loaded, as well as a count of any invalid markers that may have been in the input file. Using the wizard to load germplasms and samples, the DNA sample wizard is used to load information about germplasm, load information about samples, and link samples to DNA runs. The dataset wizard is used to load genotypic data. Again, we go through the same process where we add basic metadata, load the data file, preview the data from the file, map data fields, review and submit, and receive an email. Pane that's in green is where required information goes. There's the preview pane where you enter marker and sample positions, identify the first data point in the matrix, and map marker names and DNA run names, review, and submit. In your success email, you will see the total dataset markers and dataset DNA runs in the file. The total loaded, the total previously loaded, the already existing and the total that were invalid in your input file. You'll also see the time it took to do the load, in this case 3 minutes and 37 seconds. While loading your data set, you have the option of checking QC analysis, which will also send a success email. Once QC is completed, the QC folder will contain a number of files, one of which is a similarity matrix with Meta, an example of one. You can review this file with experts and modify and load a clean data set if necessary. Another example file from QC is the summary markers file. Here's a sample of the summary markers file with a list of the various columns in this file.